Right now, inside your body, a profound transformation is unfolding. Not from disease, not from injury, just from time. Every cell in your body carries a kind of biological clock, a counter built into the very structure of your DNA. As the years pass, that counter ticks forward. Errors accumulate. Certain systems that once ran smoothly begin to slow down. We call this aging. And for most of human history, we accepted it. The way we accept gravity, the way we accept winter. But something has changed. Scientists are now asking a question that would have seemed absurd to most researchers just 50 years ago. Not simply why we age, but whether aging is something that can, in some meaningful way, be slowed. Or even partially reversed. For most of history, we thought of aging as wear and tear, like an old car. Parts degrade. Systems fail. Eventually the whole thing stops. Aging is not one thing. It is a collection of distinct biological processes, each with its own mechanism, each interacting with the others. Scientists have now identified and named many of them. And in identifying them, something remarkable has happened. They have become potential targets. The tools that made this possible are extraordinary. We can now read a person's entire genome in a matter of hours. We can edit individual genes with molecular precision. We can observe, in real time, the interior workings of a living cell. The central question we are going to sit with throughout this video is this. If scientists now understand the mechanisms of aging in enough molecular detail, what is standing between that understanding and an actual intervention? Your body contains trillions of cells. And inside the nucleus of every single one of them sits roughly 2 meters of DNA, coiled and compressed into a space far thinner than a human hair. That DNA is the complete instruction manual for everything you are. But the DNA itself is not where aging begins. It begins, in a sense, at the very tips. At the end of every chromosome sits a structure called a telomere. Think of it as the protective plastic cap at the end of a shoelace. Every time a cell divides, those telomeres get slightly shorter, a little of the protective cap is worn away. After enough divisions, roughly 40 to 60 in a healthy human cell, the telomeres become critically short. At that point, the cell stops dividing entirely. It enters a state scientists call senescence. A senescent cell is not dead, but it is no longer doing its job properly. Worse, it begins releasing chemical signals that create inflammation in the surrounding tissue. Other cells are disrupted. These senescent cells accumulate with age. In a young body, the immune system is fairly efficient at clearing them. In an older body, fewer are removed. They build up. And that accumulation is now thought to be one of the key drivers of many age-related conditions, from joint deterioration to chronic inflammation to the loss of tissue resilience. In animal studies, scientists have shown that selectively clearing senescent cells from older mice leads to measurable improvements in physical function and, in some studies, extended healthy lifespan. Researchers are now testing drugs called senolytics in early-stage human clinical trials. These compounds are designed to selectively remove senescent cells without harming healthy ones. It is early-stage research. Human results are preliminary, but the biology underlying it is coherent and the trials are ongoing. Your actual DNA sequence changes very little as you age. What changes dramatically is something sitting on top of that code. It is called the epigenome. Think of your genome as a vast library. Millions of books, each containing instructions for a different protein or biological process. The epigenome is the librarian. It decides which books are open, which are closed, and which are available to each specific type of cell. A heart muscle cell and a neuron carry identical DNA. What makes them different is which genes are switched on. In a young cell, this system is precise. But with age, something drifts. The chemical tags on your DNA, known as methyl groups and histone modifications, begin to shift away from their original configuration. Genes that should be active get silenced. Genes that should be quiet start expressing. In 2013, researcher Steve Horvath at UCLA developed what became known as the epigenetic clock. By measuring the state of specific sites on the genome, he could calculate the biological age of a tissue. Not the chronological age, their biological age. Those two numbers can differ by a decade or more in either direction. In 2020, a research team led by David Sinclair at Harvard published findings describing exactly this kind of experiment in mice. By introducing genetic reprogramming factors into the optic nerve cells of aged mice, They were able to shift the epigenetic state of those cells toward a younger profile. Mice with age-related vision loss showed measurable improvements in visual function after the intervention. It is important to be clear about the limits of that finding. It was a mouse study, it targeted a specific tissue, it used gene therapy. We are a long way from anything comparable in humans. Your cells run on energy. That energy is produced by structures inside the cell called mitochondria. They take in oxygen and nutrients and convert them into a molecule called ATP. ATP is the universal fuel of biology. Mitochondria carry their own DNA, a remnant of their ancient origins as free-living bacteria, absorbed into more complex cells around 1 billion years ago. That mitochondrial DNA is particularly vulnerable to damage. It sits close to the site of energy production, exposed to reactive byproducts of metabolism. In a young cell, damaged mitochondria are regularly cleared away through a recycling process called mitophagy, a quality control system. As we age, that quality control system becomes less effective. Dysfunctional mitochondria persist. Energy production declines. In animal studies across multiple species, significantly reducing caloric intake while maintaining nutritional adequacy consistently extends healthy lifespan. A drug called rapamycin has shown similar effects in animal models. It targets a cellular pathway involved in nutrient sensing. It is an approved drug in humans, but for immunosuppression in transplant patients, not for aging. It carries meaningful side effects in that clinical context. Whether it could be applied safely in healthy adults is a research question, not a settled one. As we age, the immune system shifts into a state of persistent, low-level activation. Not the sharp, targeted inflammation of an infection, something quieter. It describes the chronic, low-grade inflammatory state that becomes increasingly common in older bodies. Senescent cells contribute to it by releasing inflammatory signals. Mitochondrial debris contributes to it. Epigenetic drift in immune cells contributes to it. These processes are interconnected. Each one feeds the others. The very immune system that exists to protect you becomes, over time, one of the agents of your decline. Not through failure, exactly, more through a kind of accumulated confusion. We have been treating aging as though it is simply damage, entropy, things falling apart. But some researchers argue that framing misses something important. They suggest that parts of aging are not accidental. They are, in a sense, shaped by evolution. From the perspective of natural selection, what matters is reproductive success. Once an organism has done that, there is little evolutionary incentive to keep a body running flawlessly at 90. A concept called antagonistic pleiotropy, first proposed by George Williams in 1957, suggests that genes which cause harm later in life may actually be selected for if they provide some advantage during reproductive years. A gene that sharpens immune function in your 20s might contribute to inflammatory disease in your 70s. Aging would not just be damage accumulating randomly. It would be, in part, a biological program, calibrated for a world where human life expectancy rarely exceeded 40. If aging is partly programmatic rather than purely accidental, then the question becomes, not just can we repair the damage, but can we adjust the program? There is now a substantial and growing scientific ecosystem focused specifically on the biology of aging, not as a side note of general medicine but as a primary research target. Senolytic drugs are in early human trials aimed at clearing the body of senescent cells. A pilot study published in eBiomedicine by Hickson and colleagues in 2019 tested dasatinib and quercetin. Epigenetic reprogramming is being pursued by several research groups. The question is whether it is possible to shift epigenetic state toward a younger profile without destabilizing cell identity. Still largely animal study phase. NAD is a metabolic compound that declines with age, essential for energy production and DNA repair. Compounds, including NMN and NR, are being studied. The evidence so far is mixed. Academic researchers are studying the role of specific proteins and factors in blood plasma in the aging process. The FDA has warned against commercial young blood transfusion services. The underlying research is a legitimate area of academic inquiry. There is growing interest in applying artificial intelligence and machine learning to longevity biology. Machine learning systems are being trained to identify patterns in genomic and epigenomic data. None of this is settled science. The history of gerontocins includes many promising animal findings that did not translate to people. The researchers who think most carefully about this speak not of a cure for aging, but but of a compression of morbidity, pushing serious decline into a shorter window at the end of a longer, healthier life. That is a more modest goal than immortality. But it is not a modest goal. If it becomes possible to meaningfully slow human aging, not eliminate it, but extend healthy later life by a decade or two, that would represent one of the most significant changes in the human condition in recorded history. The philosopher Martin Heidegger argued that our awareness of mortality is part of what gives life its shape. The urgency we feel, the choices we make, all colored by the knowledge that our time is limited. If that limit is substantially extended, does urgency change? Aubrey de Grey has argued that humans would adapt. That meaning does not require a fixed deadline, and that extended healthy life would create new kinds of purpose, not erase existing ones. Others are less certain. They point to social consequences. Who would have access to life-extending treatments? What happens to generational turnover when people live and hold power for much longer? What are the ecological consequences of a population that ages more slowly? There is also a biological concern specific to the brain. The accumulation of misfolded proteins, the gradual loss of synaptic connections, the vulnerability of neurons to long-term metabolic stress. A world in which physical health is extended, but cognitive decline is not straightforwardly better. In 2018, the ICD-11 included a new extension code related to aging-associated conditions, a significant step toward giving aging-related biology its own classification framework. The move was debated, and it does not represent a consensus statement that aging itself is a disease in the conventional sense. But it marked a shift in how regulatory and scientific bodies are beginning to think. Is aging inevitable? The honest answer, as of today, is that we do not know. Aging is no longer being treated as an untouchable given. It is a collection of biological processes. Those processes have names, mechanisms, and molecular addresses. Some of them have been demonstrably slowed in living organisms. A small number have been partially reversed in specific tissues in laboratory conditions. That is not the same as a cure. But it is no longer the same as nothing. There is a thought experiment in philosophy about a ship called the Ship of Theseus. Every plank of the ship is replaced, one by one, over time, until none of the original material remains. Is it still the same ship? Your body is already engaged in something similar. Most of your cells are replaced over the course of years. The atoms in your bones, your muscles, your organs are not the same atoms they were a decade ago. What longevity researchers are essentially asking is this: how far can that renewal actually go? And when we extend it deliberately, do we preserve what matters, or do we change it in ways we cannot anticipate? Those are not questions science alone can settle. They reach into philosophy, ethics, and the most personal questions any of us carry. We may not fundamentally alter the aging process within our lifetimes. But we are, for the first time, approaching that question with tools precise enough to deserve a real answer. And that is not nothing.